Welcome back to my channel Adhyan with Flow. Let's start with our new topic lag operon. In previous video we already done DNA replication, transcription, translation. Guys, in DNA replication, what does mean the DNA replicate and make their copies? In transcription, what happened? DNA split and make RNA. What about translation? That these mRNA with the help of ribosome they synthesize what? Proteins. Now we study this transcription process, like there is a DNA. DNA further divided into some segments. We call it as a promoter. We call it as a structural gene. There is some region we call it as a termination site. Okay, at the promoter side there are multiple enzymes come and bind like RNA polymerase and they transcribe these things. At the end of termination site we get complete mRNA because here RNA formation stop and here RNA formation start. So in lack of peron, in the case of bacteria, some scientists come and they identify what kind of mechanism present in bacteria, how they regulate their transcription mechanism, what kind of genes are there. Guys, in bacteria, again we call it as a there is a promoter, we can say as a regulatory unit, structural gene and termination site, same as possible. Look at here, this is the structure, here is the promoter region, here is the operator, here is their structural genes. Now I am going to discuss you one by one about each part, what kind of function they cause. Okay, first of all you have to remember two terms, activators and repressor. Guys, according to their name, activator means they active something, that's it. Listen to me, that's it. Repressor does it means they repress something, that's it. Remember these two words. If I call they work as an activator, it means they active, they start transcription. If I use this term repressor, it means they repress, it means they stop, they inhibit something, it doesn't synthesize anything. Okay, in the absence of inducer, you can also call it as an activator. In the absence of inducer, what will happen? There is a promoter, yes or not? Promoter, operator, what they do? I am going to tell you. Operator, here multiple enzyme comes and bind. They want to synthesize these structural genes ZYA. They want to synthesize these structural genes. This synthesize and make RNA. This gene synthesize and make RNA. This gene synthesize and make RNA. And further these RNA with the help of ribosome, they make proteins. The name is, this protein is beta galactosidase, this protein is permease, this protein is what? Transacetylase. Now guys, here is inducer is absent, so no one can induce this mechanism. Here no inducer, does mean here is no formation of anything. Here no formation of anything. Why? Because here is promoter. Okay, promoter is present. Now here is inhibitor also present. An inhibitor with the presence of their polymerase transcribe these things. So here repressor mRNA will form. Guys, DNA is here, and DNA what they form? RNA. What kind of RNA? mRNA. In future mRNA what they form? With the help of translation they make protein. So this segment of DNA transcribe and make mRNA. This is the repressor mRNA. Why? Because this mRNA in future they make repressor protein. So with the help of translation mechanism, what they do? They make repressor protein. And what the repressor will do? They come and bind with operator. And guys, if anyone come and block this operator region, so now further process is not done. If repressor, if someone block this operator site, this is the operator. If someone block this operator, it means there is no synthesis of Z gene, no synthesis of Y gene, no synthesis of A gene. Doesn't mean there is no mRNA of Z, there is no mRNA of Y, there is no mRNA of A. Doesn't mean, it means there is no protein of gene A, there is no protein of Y, there is no protein of Z in the absence of inducer. But guys, if inducer will come, if inducer will come, what kind of inducer they say? Lactose. That's why we call it as a lac operon. If we provide to bacteria, let's eat. Okay, you can eat lactose. Now bacteria become active, they make inducers. Now what inducers will do? Here RNA polymerase will bind and they transcribe again repressor mRNA with the help of translation mechanism they make repressor protein. But if we provide lactose in medium, these make inducer and they bind with repressor. Here inducer bind with repressor. So this repressor can't reach towards the operator. So yeah, your process still continue and they transcribe what? Z mRNA, Y mRNA, A mRNA, and further they code according to their cell. Means Z mRNA. This is the exam point of view question. What Z structural gene will do? They make beta galactosidase. What Y gene, Y structural gene will do? They make permease, so they can enter. Here they make what? Pores. In the bacteria outer cell, they make pores, so lactose easily consumed. Next is transacetylase, means they add some acetyl group. Okay, so that's all about lac operon. Whatever I teaching here, already all thing is written here. All thing is written here. Let's start again. I can repeat for you last time with the help of flow chart. Okay, now here RNA polymerase will bind and they transcribe what? Inhibitor. This region is what? Inhibitor. Inhibitor. This is the inhibitor gene. And what they do? They make with the help of, you have to write this thing, with the help of transcription mechanism. Inhibitor, what they do? They make repressor mRNA. Repressor mRNA with the help of translation process. What they will do? They make repressor proteins. DNA, here DNA, with the help of transcription they make RNA. With the help of translation they make protein. DNA is double stranded, RNA is single stranded, and they are globular size. Next is operator, and these are what? Structural genes. Now what happens? In the absence of inducers, it means there is no inducer, no one can activate this operon model. So repressor come, bind with operator. You can say block the site of operator. So no synthesis of structural gene. In the presence of what? Here in the presence of structural gene, repressor and inducer bind, and now repressor can't bind with, they can't block the operator site, and there is structural genes, they synthesize their genes. First is what they do? First they will perform what? Transcription. With the help of transcription they make mRNA. With the help of translation they make protein. With the help of transcription they make mRNA and then translation protein. With the help of transcription they make mRNA of A gene and then further protein. Okay, if you provide lactose, then this operon become active. If you don't provide lactose to bacteria, this operon will reduce, they can't active. There is a many kind of operon, guys. This is not a single operon which conduct in bacteria. Also like tryptophan operon, they also multiple operon will conduct in bacteria, but according to your syllabus, there is only lac operon. So there is genes taken code. Okay, you have to remember this diagram. In the presence of inducer, in the absence of inducer, in the presence of repressor, in the absence of repressor, they modify the question language. Okay, they modify question language, but all of the concept is this. Okay guys, so this is all about your lac operon. And now there is some long lasting question like what is Genome Project? In Genome Project, I summarize you very briefly that genome means our genes. Yeah, we know na DNA. Okay, they have reason. Recently you study na, there is no protein genes in the beads form. There is some reason. They are specified. Why? Because gene particular control characters. They control characters. They control some disease. They controls our order disorders. Okay, if human, they think that if we analyze complete gene, if we analyze complete genome project, if we, what we'll do, if we memorize all the DNA, if we sequence, like if we sequence like this DNA, they do eye color responsible, this fragment of DNA what they control height of body, this will, this region of DNA what they will, they control some kind of nose structure of particular human. So this DNA fragment what they will do, they control sickle cell anemia gene. So according, with the help of these project now, we analyze our DNA system, we sequence our DNA. But this is not a one day, okay, this is not a one day thing, okay, they launched it in 1990. Now we have proper idea about human genome project. You can also get your human genome project, like go to library and take permission, and they give you, they charge something and they give you your genome project. Guys, for this human genome project now, the overall cost is, you know, $9 billion. Guys, $9 billion. You know about the price of $1? Guys, 9 billion US dollars. That much cost that time we pay human genome project, right? There is some goals about human genome project that identify the all approximately 20,000 to 25,000 gene in human DNA. Means DNA we calculate around 20,000 to 25,000 gene we know about every genes. Determine the sequence of 3 billion chemical basis pair. Guys, you know na, base pair is what? A pair with T. You remember? Are you remember or not? Double bond, triple bond. Okay, there is the 3 billion in our DNA. There is 3 billion base pair in our DNA. Store this information in database. They store this whole information, like this much sequence of DNA code this thing, this much sequence code this thing, this much sequence of DNA code this thing. Improve tools for analysis, so we can analyze any kind of order disorder, advantage disadvantage. Simple guys, why we pay this thing? Because of human welfare. Yeah, come on guys, why our country pay this, why our earth pay this much cost? Why? Because of human welfare, that's it. Transfer related technology of other sectors such as industry. Yeah, addresses the ethical. Yeah, we take permission, legal, social issues that may arise from this. Of course guys, this is not easy. Okay, 30 years take for this project, right? In this institute, here human genome project will done. Many countries support, not support, okay, like partner like Japan, France, Germany, China, like others. All right, and this project completed in 2003. All right guys, start in this and complete in this year. Other than human, there is another thing like bacteria, yeast, C. elegans, Drosophila. We also sequence these things. Arabidopsis, this is the plant, okay, these are the plants. We also sequence their genes, okay. I know we sequence human gene project, like we sequence our genome, but we also sequence C. elegans, Drosophila and Arabidopsis. These are like we sequence their all DNA. Some methodologies, like we use some bacteria artificial chromosomes, like there is DNA of our desire and there is a vector. Vector, what they do, vector they carry fragments of DNA and they insert into some host body, and we control the function, we know the function of particular genes, that's it. Okay, how we sequence, there is maximum sequence that time, like Sanger's methods, like Maxim Gilbert method, like shotgun method, like pyro sequencing. There is multiple sequencing. With the help of these sequencing now, we sequence the DNA. We sequence the DNA. Okay. Silent features about human genome. The human genome contain this much million base pair. The average gene consists of 300. मतलब single gene, how much they contain base pair, how much? So here the estimate, like approx 3000. All right, largest genome in human now. So dystrophin, there is a protein, they contain 2.5 million. There is some order like which gene control highest base pair and which gene control lowest base pair. So highest gene, the name is dystrophin, this is a protein, they contain this much bases. Total number of gene is estimated in 3000, of course 24,000 to 30,000, much lower than previous. Almost all 99% nucleotide bases are exactly same in all people, means 99.9% as same, 0.1% is different. 99.9% our gene is same, our DNA is same, only 1% is different. The function are unknown for over 50% of the discovered genes, means there we know 50% genes function, but now we know almost every function of genes. Less than 2% of DNA code for protein, 100% DNA, but only these DNA, only 2% part of DNA contribute in protein formation. Rest of the take another work. Yeah, DNA don't have any only one function, like they have only one function, then they transcribe and then translate and they make protein. They have another function also. Okay, repetitive sequence make very large portion of, guys, repetitive sequence means what? Like ATGC, 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 ATGC means something is repeated again and again and again and again. So there is very large proportion in DNA. Like there is not like a random sequence. There is not random like ATGC, ACGC, TGAC, GAC, GTAC. No, there is repetitive sequences more compared than random sequence. Chromosome has one most gene. Like they said that we have 23 pairs of chromosome. So chromosome number 1 now they have highest genes, and chromosome Y now in male they have fewest genes. That's it. Scientists have identified about 1.4 million locations where single base DNA difference, single nucleotide polymorphism means, guys, nothing is different. Single nucleotide polymorphism is what? Like some human have ATG sequence, another this sequence. Single, only single, only single. Look at here, only single. There is only single base pair is different, rest of these are same, rest of these are same. These are only if only single base pair is different, but rest of the all is different. Means I take this is the human first, this is the human second, this is the human third, fourth, fifth, sixth, seventh. What I identify that their sequence is starting sequence and ending sequence is same, but the middle part of the sequence is different. That's it. But rest of the all is same. This is the human first. They have also ATG. Human 2 they have AG, but in the middle part they have A. So there is single nucleotide polymorphism. If in person to person only one nucleotide difference. Okay. Now okay, this is what fingerprinting mechanism. In fingerprinting mechanism now is easy. Now is easy. This technique based to differentiate human to human. Like your gene 99.9% same, my gene is 99.9% is same. Now another person is 99.9% is same. Now how we calculate that this is the father, this is the mother, and this is the son. Like how we trust that this son is for these parents? Because guys, because if we take father's DNA, if we take mother DNA, and if we take son's DNA, and we do DNA gel electrophoresis, so we get bands. Whatever we bands we get from the father, whatever band we get from mother, they are different now because father is different. They do their different features. Like we know 99.9% their DNA, this DNA or this DNA is same. But how we conclude that yeah, this is the son of this father and mother? They have this kind of arrangement. So now, can you identify? Can you identify? Yeah, because some genetic balance is present in this cell. So you can easily identify. If you run, like this is the mother first, this is the mother second, and mother third. These mother fight for this daughter, that this is my daughter. How we calculate, how we identify that this is the daughter of this particular. We also use in the crime scene also. Okay, don't panic. This band is this and this. This band we get from mother first. Okay, this band we get from mother 2, and this band we get from mother third. And now when we take the DNA of this daughter, what kind of band we get? How we calculate this the daughter and which is here real mother? We get this type of band. Now tell me, when I run this daughter DNA, when I run this mother DNA, this mother, tell me what you watch, that her band match with which mother? Right guys, mother second. These DNA is none other than, these are the satellite DNA. We take only small repetitive sequence, we call it as a V and TR. So this technique is very useful. How we calculate, like guys, take some DNA samples, it may be some crime scene, it may be their blood sample, it may be their semen sample, it may be their hair, nails, whatever. Okay, now here we take agar. Agar is what, there is a, this is the algae, this is the algae. Okay, we get it from the laboratory in the powder form. Okay, take this agar in this beaker size, add some distilled water and heat. No one tell you this technique. Heat and wait. So this powdery form of agar will dissolve in this water medium. This is not a normal, okay, this is the buffer medium, okay, this is the not a normal water, okay, distilled water. Okay, now here is the kit of gel electrophoresis kit. Here there is a comb is here, they make holes. Okay, now after transparency of this gel, this solution we pour into this, we pour in this. After pouring now, they become solidified. If we wait, still wash, they become solidified gel. They make a transparent solidified gel. Now what they do? They pour this, and this, and this, and now connect with some light apparatus, anode, and connect the electricity. And here what happen? DNA will travel. DNA will travel throughout this DNA, and you can calculate who is the real mother of daughter and mother. Clear or not? Clear or not? Guys, these gel are formation like this manner. If DNA is large in size, they blocked. If small in size, they, and very small size, they reach forward. Heavy DNA block here. Okay, heavy DNA block here, but woo not heavy, they still continue forward, they move forward. So we get bands in this manner. So we can estimate this is the heavy, like assume this is the 100 kilodalton weight DNA, this is the 80 kilodalton weight, this is the weight of 70 kilodalton, this is the weight of 60, 50, 40. Okay, because here we have scales, so we can measure this is a 100 kilodalton band. Here if I got band, then this is the 90 kilodalton. We estimate na liter, kilometer, millimeter. So according to DNA na we measure in kilodalton. So this is all about DNA fingerprinting. Look at here, there is a step. There is a step. Isolation of DNA. This is normal, not normal like we can isolate the DNA easily. No, we have to provide some DNA. So they break this DNA. We get small fragment. Then we collect these fragment. And now what we'll do? We need probe. What is probe? There is no artificial we made in laboratory. Some sequence of RNA and DNA. RNA and DNA. Alright or not? And we probe and labeled with VNTR probe, means if we get fluorescence, so assume when this DNA is run, so they spark here, so we can visualize properly. We can also labeled with ETBR. In laboratory na ETBR is present, so here we got a band, so this band is not visible. We take this gel into the spectrophotometry, UV spectrophotometry, and this is harmful. So we take this electrospectrometry, so we can visualize these band. Some you can watch on YouTube also, you can easily understand. If you go and search na gel electrophoresis, still my video also on DNA gel electrophoresis, you can watch and you can betterly understand about DNA fingerprinting. Okay, this is the very shortcut, I teach you in detail. Okay, this is the process mechanism. Look at here, there is a comb place. Clear guys? And I think your chapter is complete. Yeah, so we will meet in our next video with a new chapter. That's all for now. Thank you guys, keep smiling and don't forget to subscribe my channel Adhyan with Flow. Thank you so much for listening me, giving me such a time. Thank you so much.